Hello friends and welcome. Today we are going to talk about reproduction in bacteria. The main objectives of today's deliberation are to give a brief idea about bacteria. Second, to find out different types of reproduction in bacteria. Third, to know about the asexual methods of reproduction in bacteria. And finally, to give a detailed insight about sexual reproduction in bacteria. To begin with, let me give a brief introduction. The production of new individuals by the existing ones is called reproduction. There are two quite distinct methods of producing offsprings, viz. Asexual and sexual methods. The asexual reproduction involves a single parent and produces offsprings which are genetically identical to the parent. The sexual reproduction involves genetic recombination between two parents and so produces offsprings which differ not only from the parent but also from each other. Unlike in multicellular organisms, increases in cell size, that is cell growth, and reproduction by cell division are tightly linked in unicellular organisms. Bacteria grow to a fixed size and then reproduce through binary fission, a form of asexual reproduction. Under optimal conditions, bacteria can grow and divide extremely rapidly, and bacterial populations can double as quickly as every 9.8 minutes. In cell division, two identical clone daughter cells are produced. Some bacteria, while still reproducing asexually, form more complex reproductive structures that help disperse the newly formed daughter cells. Examples include fruiting body formation by myxobacteria and aerial hyphae formation by streptomyces or by budding. Budding involves a cell forming a protrusion that breaks away and produces a daughter cell. In the laboratory, bacteria are usually grown using solid or liquid media. Solid growth media such as agar plates are used to isolate pure cultures of a bacterial strain. However, liquid growth media are used when measurement of growth or large volumes of cells are required. Growth in stirred liquid medium occurs as an even cell suspension, making the cultures easy to divide and transfer. Although isolating single bacteria from liquid media is difficult. The use of selective media, that's media with specific nutrients added or deficient or with antibiotics added, can help identify specific organisms. Now, let's talk about types of reproduction in bacteria. Bacteria can't reproduce by mitosis or meiosis because they have no nucleus. Instead, they have evolved different methods of reproduction. Bacteria reproduce both asexually and sexually, that is, genetic recombination. First of all, let us discuss asexual reproduction in bacteria. Asexual reproduction in bacteria occurs by the following methods. Number 1, binary fission. This is the most common type of asexual reproduction in actively growing bacteria and occurs during favorable conditions. On this basis, bacteria were once called fission fungi, that is, schizomycetes. In this process, the cytoplasm and the nucleoid divide equally into two without mitosis, and the two daughter cells formed are identical to each other. Hence the name binary fission. The whole process of binary fission involves two steps: genome replication and septum formation. Both of the events occur simultaneously and are triggered by a mesosome if present. First, genome or DNA replication. Binary fission begins with DNA replication. DNA replication starts from an origin of replication, which opens up into a bubble. The replication bubble separates the two DNA strands. Each strand acts as a template for synthesis of a daughter strand. The DNA replication is bidirectional. Starting from the point of origin and resulting in the formation of two circular daughter DNA molecules. In each daughter DNA molecule, one strand is derived from the parental DNA molecule, while another strand is a new one. This is semi-conservative mode of DNA replication. Now, septum formation or cell division. A peripheral ring of plasma membrane invaginates and grows centripetally to form a double membranous septum. Wall material is deposited between the two membranes of the septum. This separates the parent cell into two nearly equal daughter cells, each having its own nucleoid. Under optimal conditions of nutrition, water, and temperature, the process of binary fission is very quick and the division may be completed in about 20 to 30 minutes. Thus, in 24 hours, a very large number of bacteria may be produced. Second, zooglia stage. Under unfavorable conditions, the bacterial cells come to lie in chains and get surrounded by a lot of mucilage. This is called zooglia stage. It is a temporary phase for avoiding desiccation. Third, conidia formation. This condition occurs in mycelial bacteria. The terminal portions of bacterial filaments cut off rounded structures in chains called conidia. These conidia on detachment form new individuals. Number 4, gonidia formation. In this case, the bacterial cell produces a number of flagellated daughter cells within. These daughter cells are also called as swarmers. The cell wall of parent cell ruptures, releasing gonidia. This is reported in Rhizobium species. Number 5, cyst formation. This is reported in Azotobacter. The parent cell secretes a highly protective covering called a cyst wall, which can resist unfavorable conditions. On return of favorable conditions, the cyst wall dissolves and the bacterium gets released. Thus, cyst formation helps in perennation. Number 6, budding. Some bacteria continuously produce protrusions called buds, which on detachment form new individuals. Hyphomicrobium vulgare and Rhodomicrobium vanillii are common examples of budding bacteria. Number 7, fragmentation. It occurs in colonial cyanobacteria. After reaching a certain length, the blue bacterium breaks up into pieces called fragments. Each fragment is the beginning of a new colony. Number 8, harmogonia. It is a characteristic method of reproduction in blue-green algae. Trichomes of the filamentous genera break up within the sheath into short sections or segments of one to many uniform living cells. These short segments of trichome are called harmogonia or harmogones. The harmogonia may be separated by number 1, the formation of heterocyst, for example in nostoc. Second, the death of one or more cells, for example oscillatoria. Number 9, echinates. These are formed in many cyanobacteria. Echinates are thick-walled, resistant cells with abundant food reserves which are frequently developed singly next to heterocyst. However, their position is variable, for example, anabina. Number 10, harmospores. They are formed at the tip of trichomes or short side branches of cyanobacteria, for example, Vestilia. Number 11, exospores. In certain blue-green bacteria, the spores are successively cut off at the distal end of the protoplast by transverse divisions. These are called as exospores. Number 12, endospore formation. When the environmental conditions are adverse, some gram-positive bacteria, especially bacilli, and certain blue-green bacteria, for example Stichosiphon, produce thick-walled resistant spores called endospores. Endospores are formed under conditions of nutrient deficiency, unfavorable temperature, presence of toxic substance, etc. Iron and manganese stimulate their formation. Flagella, if present, are withdrawn. Endospores can be formed in middle, subterminal or terminal of the sporangial cell. The endospores can be rounded, oval or cylindrical in outline. During endospore formation, a portion of cytoplasm and a copy of bacterial chromosome dehydrate and get encased by a very thick wall. The rest of the cytoplasm and the bacterial cell wall deteriorate so that the endospore is released. The thick wall of a typical endospore is distinguishable into four parts, namely exosporium, spore coat, cortex, and core wall. Exosporium is a loose outer covering which is present only in some cases. Spore coat is hard and resistant. It is made up of keratin-like disulfide-rich proteins. Cortex is a thick layer formed of mucopeptides and carbohydrates. Core wall is rich in proteins. It is also called inner cortex. The protoplasm of the endospore has two parts, cytoplasm and nuclear body. The cytoplasm is in a highly dehydrated state. About 90% of it is made up of proteins. It is also rich in lipids, calcium, and manganese, but is deficient in potassium and phosphorus. Endospores contain the anticoagulant dipicolinic acid, which makes them highly resistant. Now, germination of endospores. The endospore can remain dormant for several decades and can tolerate hardest of environments, desert heat and dehydration, boiling temperature, polar ice, toxic chemicals, and even extreme UV radiations. They germinate only under favorable conditions. The protoplast absorbs water and swells up. Dipicolinic acid leaks out so that the protoplast becomes active. The swollen protoplast breaks the spore covering either at the equator or at one end. It comes out as a new bacterium surrounded by the thin core wall. Now, resistance of endospores. Endospores are not affected by toxic chemicals and exposure to high 60 to 70 degrees Celsius or low -100 degrees Celsius temperatures. Some of the endospores are not killed even in liquid helium -269 degrees Celsius. The endospore of anthrax bacteria can withstand dry heat of 140 degrees Celsius for 3 hours. This extreme resistance of endospores is due to first, very low water content, second, anticoagulant, that is dipicolinic acid, third, thick, resistant and impermeable covering, fourth, lower rate of metabolism, fifth, absence of active enzymes, sixth, deficiency of potassium and phosphorus, and seventh, increased calcium content. Fortunately, most bacteria do not produce endospores, the only exceptions being tetanus and anthrax bacteria. An uncommon type of food poisoning called botulism is caused by the germination of endospores of Clostridium botulinum inside the food. Now, coming to second type of bacterial reproduction, that is sexual reproduction or genetic recombination in bacteria. Cytological observations and genetic studies indicate that something like sexual reproduction involving the fusion of two different cells and a transfer of hereditary vectors occurs in bacteria, although infrequently. But typical sexual reproduction through the agency of gametes is absent in bacteria. There is no fertilization and meiosis. However, the gene transfer in bacteria occurs by three methods: conjugation, transformation, and transduction. Number 1. Conjugation involves transfer of DNA from donor or male cell to a recipient or female cell through a specialized sex pilus or conjugation tube. Second, transduction involves transfer of bacterial genes from a donor cell to a recipient cell by a bacteriophage. Third, transformation. Transformation involves the uptake of naked DNA molecules from one bacterium, the donor cell, by another bacterium, the recipient cell. Therefore, conjugation occurs through direct cell-to-cell contact, but transformation and transduction do not involve any such contact. The transfer of genetic information is thus a one-way transfer rather than a reciprocal exchange of the genetic material. The phenomenon has been called parasexuality or genetic recombination. However, sexual reproduction or even conjugation is unknown in cyanobacteria. Now, let us discuss these three processes in detail. Number 1. Conjugation. A mechanism resembling sexual reproduction was discovered by Joshua Lederberg and Edward Totem. They are both from USA in 1946 in Bacterium E. coli. It was confirmed by Hayes from London and Wollman from Paris in the same bacterium. They found that physical contact was involved in conjugation between the two conjugates and that DNA from one is transferred into another through a conjugation tube. Therefore, bacterial conjugation may be defined as a transfer of genetic material from a donor cell to a recipient cell through a specialized intercellular connection or conjugation tube that forms between them. The donor and the recipient cells are sometimes referred to as male and female cells respectively. Now let's talk about mechanism of conjugation. The cells that have the capacity to serve as donors during conjugation are differentiated by the presence of specialized cell surface appendages called F-pili. The synthesis of these F-pili is controlled by several genes that are carried by small circular molecule of DNA of about 100 kilobase pairs called an F factor for fertility factor, or also called as F plasmid or sex factor. The F plasmid is an episome, a plasmid that exists in two different states. Number 1, the autonomous state, in which it replicates independently of the host chromosome, and second, the integrated state, in which it is covalently inserted into the host chromosome and replicates along with the host chromosome, like any other set of chromosomal genes. It carries its own origin of replication, the ORIV, as well as an origin of transfer or ORIT. There can only be one copy of the F plasmid in a given bacterium, either free or integrated, two immediately before cell division. The donor bacteria carrying an F factor form the conjugation tube and are called F positive or F plus, denoted as F plus. Strains that lack F plasmids are called F negative or F minus and are denoted as F minus. When conjugation is initiated via a mating signal, a relaxase enzyme creates a nick in one plasmid DNA strand at the origin of transfer, or ORIT. The relaxase may work alone or in a complex of over a dozen proteins known collectively as relaxosome. The transferred or T strand is unwound from the duplex plasmid and transferred into the recipient bacterium in a 5' terminus to 3' terminus direction. The remaining strand is replicated either independent of conjugative action, that is vegetative replication, beginning at the ORIV, or in concert with conjugation, that is conjugative replication, similar to the rolling circle replication of lambda phage. Conjugative replication may necessitate a second nick before successful transfer can occur. In bacteria, conjugation can occur in several ways. Some important examples are as follows. Number 1, conjugation between F plus male and F minus female. E. coli shows two strains, one acting as donor, that's F plus male, and other as recipient, F minus female. The donor cell contains the F factor in the autonomous, that is F plus cell, conjugates with an F minus recipient cell, only the F factor is transferred. The fertility factor is usually accompanied by the presence of pili. They help the donor cell to get attached to the recipient cell. In the region of contact, F plus grows in size and produces a conjugation tube. F factor replicates. A copy of it gets transferred to the recipient cell, which also becomes donor. Thus, mixing a population of F plus cells with a population of F minus cells results virtually in all the cells in the new population becoming F plus. In other words, the sex in E. coli can be called infusious. Second, conjugation between HFR male and F minus female. The F factor can integrate into the host chromosome. An F plus cell carrying an integrated F factor is called an HFR, that's for high frequency recombination. Therefore, the F plus male becomes HFR male. In the integrated state, the F factor mediates the transfer of a chromosome of the HFR male cell to a recipient, that is F minus cell. Usually, only a portion of the HFR chromosome is transferred before the cells separate, thus breaking the chromosome. Only rarely will an entire HFR chromosome be transferred. The mechanism of transfer of DNA from a donor to a recipient cell during conjugation appears to be the same, whether just the F factor is being transferred as in F plus by F minus matings, or the chromosome is being transferred as in HFR by F minus matings. Transfer is believed to be initiated by an endonucleolytic nick in one strand at a specific site, which is the origin of transfer on the F factor. The 5' end of the nicked strand is then transferred through the conjugation tube into the recipient cell. Transfer is believed to be coupled to rolling circle replication, with the intact circular strand being replicated in the donor cell and the displaced strand being replicated in the recipient cell as it is transferred. Because the origin of transfer is within the integrated F factor, one portion of the F factor is transferred from an HFR cell to an F minus cell prior to the sequential transfer of chromosomal genes. The remaining part of the F factor, however, is the last segment of DNA to be transferred. Thus, in HFR by F minus matings, the recipient F minus cell acquires a complete F factor, that is, becoming an HFR donor only in those rare cases when an entire HFR chromosome with its integrated F factor is transferred. Third, gene transfer by F' factor. The integration of the F factor into the bacterial chromosome is reversible. The F factors can be excised from the chromosomes so that the HFR cell reverts to an F plus cell. This excision occurs at about the same frequency as integration. Correct excision depends upon a break occurring at the same site on the chromosome as the integration site. In rare cases, however, the break occurs at a neighboring site, so that on excision, a neighboring segment of the host DNA remains attached to the F factor. Such an F factor containing a small piece of chromosomal DNA is called an F prime factor. The origin of a F prime factor is analogous to the formation of specific transducing phage. The cell containing an F prime factor is called a primary F prime cell. The DNA integrated into the F prime factor can now be transferred from the F prime donor cell to an F minus recipient cell with the same frequency, that is 100%, as the F factor from F plus strains to F minus strains. The same piece of chromosomal DNA would be transferred from HFR cells to F minus cells only with a frequency of 1%. Transfer of the F prime factor from a primary F prime cell in which it originated to normal F minus cell results in a secondary F prime cell. In this cell, the segment of the bacterial chromosome is present twice, that is in the diploid state, resulting in the formation of partial diploids or merozygotes. Recombination of this type mediated by F prime factors is called sexduction or F-duction. Because of the partial diploidy resulting from sexduction, it provides an important method for determining dominance, relationships between alleles and defining genes by complementation tests in bacteria. Now coming to second type of sexual reproduction, that's transduction. It was first discovered by Ann Zinder and J. Lederberg in 1952 in Salmonella typhimurium, a mouse typhoid bacterium. Transduction is the transfer of DNA from a donor cell to a recipient cell by bacteriophages. In most cases, only a small segment of the host, that's the donor DNA, is transferred. Now coming to lytic and lysogenic cycles. Transduction happens through either the lytic cycle or the lysogenic cycle. Based on their interactions within the bacterial cell, bacteriophages are classified into two types: virulent phage, which always multiply and lyse the host cell, and temperate phages, which have a choice between two lifestyles after infection. They can either enter the lytic cycle, during which they reproduce and lyse their host cells just like virulent phages, or alternatively, they can enter the lysogenic pathway, during which the phage chromosome is integrated into the bacterial chromosome, where it can remain dormant for thousands of generations and replicate like any other segment of the host chromosome. If the lysogen is induced, for example by UV light, the phage genome is excised from the bacterial chromosome and initiates the lytic cycle, which culminates in lysis of the cell and the release of phage particles. The lytic cycle leads to the production of new phage particles, which are released by the lysis of the host. In both cases, the transducing phages are usually defective in some respect. For example, they often lose the ability to lyse host cells. Now let us discuss types of transduction. Two kinds of transduction can be distinguished: generalized transduction, which can transfer any part of the host DNA, and specialized transduction, which is restricted to the transfer of specific DNA segments. In certain cases, bacterial DNA is injected by a phage, but it does not replicate. This kind of transduction is referred to as abortive transduction. Now let's discuss these two types of transduction in detail. Generalized transduction. In generalized transduction, a random or nearly random segment of bacterial DNA is wrapped up during phage maturation in place of or along with the phage chromosome in a few progeny particles called transducing particles. Generalized transducing phages can therefore transport any gene of the donor cell to the recipient cell. Since all the genes of the donor are represented in a population of these transducing particles, This type of transduction was named generalized transduction. In some cases, generalized transducing particles contain only bacterial DNA, and in other cases, they contain both phage and bacterial DNA. Generalized transduction is mediated by some virulent bacteriophages and by certain temperate bacteriophages whose chromosomes are not integrated at specified attachment sites on the host chromosome. Generalized transducing particles are produced during the lytic cycles of these phages. Number 2, specialized transduction. In specialized transduction, a recombination event involving the host chromosome and the phage chromosome occurs, producing a phage chromosome containing a segment of bacterial DNA. Specialized transducing particles thus always contain both phage and bacterial DNA. Specialized transduction is so named because a given virus transduces only genetic markers of the host that are located in one small region of the bacterial chromosome. Specialized transduction is mediated by the temperate bacteriophages whose chromosomes are able to integrate at one or at few specified attachment sites on the host chromosome. In its integrated state, the phage chromosome is called a prophage. The chromosomes of temperate phages of this type are thus capable of both autonomous replication, that's replication independent of the replication of the host chromosome, and integrated replication, that is replication as a segment of the host chromosome. As such, they are examples of genetic elements called episomes. And finally, let's talk about third type of sexual reproduction, which is transformation. Frederick Griffith in 1928, an English bacteriologist, accidentally found that the heat-killed bacteria of virulent strain of Pneumococcus pneumoniae could transfer characteristics of its strain to the non-virulent strain of living bacteria. Avery, MacLeod, and McCarthy in 1944 observed that it is due to the transfer of DNA segments from the dead cells to the living cells. They called the uptake and incorporation of DNA by bacteria as transformation and the normal ability to take up exogenous DNA from the environment as competence. Therefore, transformation may be defined as the gene transfer by soluble or naked DNA which has been extracted or otherwise liberated from a donor bacterium to a recipient bacterium. This is the longest known and historically the most important kind of gene transfer in bacteria. Natural transformation is a bacterial adaptation for DNA transfer that depends on the expression of numerous bacterial genes whose products appear to be responsible for this process. In general, transformation is a complex, energy-requiring developmental process. In order for a bacterium to bind, take up and recombine exogenous DNA into its chromosome, it must become competent, that is, enter a special physiological state. Competence development in Bacillus subtilis requires expression of about 40 genes. The DNA integrated into the host chromosome is usually, but with rare exceptions, derived from another bacterium of the same species and is thus homologous to the resident chromosome. In Bacillus subtilis, the length of the transferred DNA is greater than 1271 kilobase pair, that is more than 1 million bases. The length transferred is likely double-stranded DNA and is often more than a third of the total chromosome length of 4215 kilobase pairs. It appears that about 7 to 9% of the recipient cells take up an entire chromosome. The capacity for natural transformation appears to occur in a number of prokaryotes, and thus far 67 prokaryotic species in 7 different phyla are known to undergo this process. Competence for transformation is typically induced by high cell density and/or nutritional limitation, conditions associated with the stationary phase of bacterial growth. Transformation in Haemophilus influenzae occurs most efficiently at the end of exponential growth as bacterial growth approaches stationary phase. Transformation in Streptococcus mutans as well as in many other streptococci occurs at high cell density and is associated with biofilm formation. Competence in Bacillus subtilis is induced towards the end of logarithmic growth, especially under conditions of amino acid limitation. By releasing intact host and plasmid DNA, certain bacteriophages are thought to contribute to transformation. With this, we conclude today's lecture. Hope this was helpful. Thank you and goodbye.